And go anytime you're ready.
Well, greetings and salutations. Good afternoon, everyone, or good morning if you happen to be in that part of the world, or perhaps good evening. Welcome to the 11th installment of the 21st year in 2022 of the Demystifying Medicine series from the NIH. I'm Dan Kastner. I am the Scientific Director Emeritus of the National Human Genome Research Institute, and I'm the co-chair of the Demystifying Medicine series this year with founder and long-term single host, Win Arias, who's actually joining us with the bow tie, waving to all. So in any case, the Demystifying Medicine series is fantastic. The goal of the series is to bridge exciting developments in biology and engineering with medicine. Our sessions are on Tuesday afternoons from 4 until 6 p.m. Eastern Standard Time. Now it's Eastern Daylight Time, from January 4th to May the 3rd. And we're videocast at the link shown on this slide. Our course schedule is also shown a link there. We go until, I guess, the 3rd of May, if we're fortunate. If you attend at least 50% of sessions and pass a final exam of fairly easy questions, you will receive a certificate, a CME certificate. And the number that you need to copy down right now, if you want CME credit, is 38651. Again, that's 38651. So in any case, if you have questions during the course of the presentations today, please submit them as you think of them by using the Send Live Feedback icon on the videocast display. All of the previous sessions are archived at the link that's shown there, going back the full almost 21 years of the series. So you can feast on it if you so desire. And for additional information, you can email Dr. Arias at the email address shown here. Rita, if we can have the next slide, you can see here the iconic Brooklyn Bridge going from the borough of Manhattan. It wasn't a borough at the time. I think it was a separate city, to the city of Brooklyn, being built in the 1870s and early 1880s. And it is a bridge that brings together two distinct places. And the whole idea is that by building bridges, we can exchange ideas and we can expand our horizons, because of course nobody knows everything about everything. And you see here on the catwalk on the left, these two guys who are there at the construction of the Brooklyn Bridge, one of them being a basic scientist from Manhattan, and the other being a clinician from Brooklyn. And they are there talking about various medical topics of the day, including questions about the pathophysiology of various infectious diseases that were common at the time. If we can go to the next slide, we have the introduction of the first of our two speakers today. So our first talk, which will begin after I'm done with these introductions, is entitled Somatic Mutations in Benign Diseases. And I think that many of you know that somatic mutations, that is to say, variants in the genome that arise after the birth of the organisms, that happen in the somatic cells of the body, that they can be responsible for cancerous disease, malignant diseases. But it turns out that there are somatic mutations that also underlie benign, non-malignant diseases, non-cancerous diseases. And this is a new insight that has come about over the last few years. And the person who will introduce us to this new paradigm in thinking is Dr. Neal Young. He is a senior investigator and chief of the cell biology section, and also chief of the hematology branch of the National Heart, Lung, and Blood Institute in the intramural program of the NIH. Dr. Young got his bachelor's degree, cum laude, from Harvard College, and then his medical degree from Johns Hopkins. He then went on to do postgraduate training at the Massachusetts General Hospital and at Barnes Hospital in St. Louis. He is a master of the American College of Physicians and a member of both the American Federation for Clinical Research and the Association for American Physicians. He's the recipient of the Samuel J. Heyman Service to America Medal in Science and Environment. His research interests are very broad and focused actually on some of the topics that we're going to be talking about today. First of all, earlier in his career, he developed a treatment for aplastic anemia. It was the first multi-center clinical trial using anti-thymocyte globulin, ATG. And he helped establish aplastic anemia as an immune-mediated disease by virtue of the beneficial response to ATG. ATG is now the standard of care for treatment of aplastic anemia, with 1 year survival having increased from 10% to over 80%. And patients with aplastic anemia owe a great debt of gratitude to Dr. Young. In addition, he's had an interest in parvovirus B19 infection and hematopoietic cells, and he discovered the role of mutations in the telomerase gene, TERT, in aplastic anemia, acute myeloid leukemia, and hepatic cirrhosis. If I can have the next slide, I will introduce our second speaker, who is the good Dr. Peter Grayson. So Dr. Grayson is going to be speaking about somatic mutations in rheumatologic diseases, and he is going to take us to the state of VEXAS, wherever that is, and beyond. Dr. Grayson is an Earl Statman investigator and chief of the Vasculitis Translational Research Program at the National Institute of Arthritis and Musculoskeletal and Skin Diseases. And he's also a mensch. He is the acting director of the NIAMS Rheumatology Fellowship Program, having stepped in when the previous director had retired until the program can identify a permanent director. But here, Dr. Grayson is on the tenure track, and he's doing this great service to the NIH, and my hat is off to him. He got his bachelor's degree at Brown University and his medical degree at the Medical University of South Carolina. And he then got a Master of Sciences degree at Boston University and did postgraduate clinical training in internal medicine and rheumatology at Boston University and a vasculitis fellowship from the legendary Vasculitis Clinical Research Consortium. When he was a fellow in rheumatology, he was a recipient of the American College of Rheumatology Distinguished Fellow Award. His interests are in the natural history, biomarker discovery, genetics and genomics, molecular classification, advanced imaging, and clinical trials in the many forms of systemic vasculitis. He also has an interest in novel pathways of neutrophil-mediated inflammation in monogenic vasculitis, and he is the senior author on a New England Journal of Medicine paper that was published on New Year's Eve of 2020, right at the end of 2020, defining VEXAS and raising the possibility that somatic mutation plays an important role in adult onset rheumatic disease, molecularly defining new disorders that cut across clinically based taxonomy. That's a mouthful. So in any case, without further ado, I would like to turn things over to the good Dr. Young, who will regale us with stories of somatic mutation in benign disease. Dr. Young, please take it away.
Okay. Dan, thank you for that nice introduction, although I also would like to be called a mensch. And as Dan said, I'm going to talk about mutations that are acquired during life, not born with them. You don't pass them on to your children. that cause benign disease. And as you'll hear from Peter's interview with the patient, these are really not benign in the sense that they're harmless or pleasant in any way. They're very serious diseases, but they're not cancer. Now I'm going to deal with a lot of material, and I'm going to try to bridge also the gap between lay people who have an interest in this new class of diseases and those who are more sophisticated. So I'm going to summarize some of the more complicated slides that I have that I hope make them comprehensible to everyone. Here are my financial disclosures. These are just agreements between NIH and industry for research. And these are the learning objectives. I want you to understand somatic mutations and some of the diseases, and especially to understand that Darwin is still quite applicable and maybe even will infuse us with better understanding of what's happening with this new area of acquired mutations as causative of disease. Now, to some extent, my talk will be an exegesis of a review that we also published just within the last couple of years with my colleague and close friend, Satu Masjoki, who leads an institute at the University of Helsinki. This is a pandemic project that we did virtually, and I think made both of our lives much easier during this difficult period. And the idea of this review, because we didn't invent the idea of somatic mutations causing these diseases, nor are we necessarily the major contributors, was that we thought that they were not really very well appreciated as a class of disease, a new class of diseases. And part of that has to do also with Dan and Peter, with whom we've collaborated on VEXAS. You'll hear about VEXAS in detail from Peter. But when we understood that VEXAS was a disease, a somatic mutation disease in hematopoietic cells, as smart as they were, brilliant as they were, they didn't know anything about somatic mutations at all. And I'm a hematologist, and I know somatic mutations quite well. And I'm going to tell you that introduction. So the genome first discovery of VEXAS, which as Dan indicated, was an NIH contribution to this area, and a really important one in terms of patients. We in hematology, not to sound too snooty, we've known about somatic mutations as causing disease since the late 19th century, and a case report by a quite famous and rich English physician named William Gull. So I'm going to tell you mainly about PNH in my introduction and leave it to Peter to talk about VEXAS. Now, I'm a hematologist, so I'm even interested in somatic mutations or genetics or diseases that go beyond the blood. Really has its root in this not good clinical scenario. This is a photo of a patient with now what we understand to be immunologically mediated aplastic anemia, who had terrible blood counts, bleeding, infected, and anemic. An empty bone marrow is shown below. And we treated her with this regimen that Dan referred to, immunosuppression, and really quite miraculously, her blood counts recovered, saw her in clinic, hugs every year, doing fine with almost normal blood counts. And then 10 years later comes back with a bone marrow that doesn't show fat, but actually shows leukemic type cells, chromosome abnormalities, and she's undergone what we call in bone marrow failure, malignant clonal evolution, a very bad thing for the patient, very demoralizing as well for the physicians. And clonality, acquired clonality, is quite familiar to hematologists, especially in the area of marrow failure, my area of expertise. And I'm going to tell you about PNH, but we have other mutations that occur in stem cells, some of which are quite bad, like the chromosome abnormalities and also somatic mutations that lead to malignant or pre-malignant conditions, some of them seemingly positive because they allow stem cells to escape from the immune system, and of course, PNH. We have mutations that occur in aplastic anemia in a subset of genes that are actually known for other reasons. The name of these genes is not important, but they occur in a quite substantial number of our patients, don't lead to cancer in most individuals, but they're shared as genes that are mutated with a large portion of the population. This is a brief description of a phenomena. It's not really a disease, but it's a finding in older individuals of clonal hematopoiesis, meaning that there are white cells in the peripheral blood and precursor cells in the bone marrow that are mutated in a specific set of genes that are also mutated in leukemia or in the pre-leukemic condition of myelodysplastic syndrome. Now, if you're an older person like me, this is really bothersome because it's not rare. And on the order of 25 or 30% of people who are above the age of 70 or 80 will show on fairly simple clinical genomic testing evidence of a mutation in one of the genes that I just showed you also is mutated in aplastic anemia. How predictive these are of leukemia is still a bit unclear, but the associations that have been made with some of these mutated cells, mutated clones, have been with diseases outside of the hematologic system and not with cancer, but for example, with atherosclerotic disease, coronary disease, and stroke, more recently with chronic obstructive pulmonary disease. Which is an extraordinary disease. As I said, it's been known since the mid-19th century, and partly came to the attention of Sir William Gull because it has a very striking clinical manifestation, which is that patients can classically wake in the morning and their urine is quite dark. I'm showing you some examples on the left. Actually, there's a grading system, sort of like going to the hardware store, of how much blood cell destruction has occurred because their urine is dark, not because it has red blood cells. That's the typical reason that people become alarmed if they see a blood in their urine at any time, but they have hemoglobinuria, not actual red cells, but the pigment within the red cells. And that's due to the fact that their red cells are lysing, that they're popping actually in the circulation, which is not very common actually as a hemolytic anemia syndrome. Most of the time, the red cells are removed by the liver and spleen. The paroxysmal nocturnal hemoglobinuria is the name, but actually it's not the hemolytic anemia that most affects patients or actually kills them. And they have other very striking distant manifestations. Immune aplastic anemia I've mentioned, and you'll see that the association is very strong with the disease that I study. But what kills most patients and can be absolutely catastrophic are very large blood clots that occur, thromboses that occur in the venous system and can actually destroy an organ over the course of just a couple of days. So lots of different things that can happen with PNH. I've shown you also some of the symptoms and signs that can be seen. Very mysterious because this is a genetic disease that's acquired. Now, the origin of PNH has now been worked out by some truly brilliant geneticists and hematologists. It was very baffling at the outset because PNH was worked out over the course of 150 years to be a disease in which the red blood cells were sensitive to complement. And also it was worked out that the red blood cells lacked a lot of their membrane proteins. And it was worked out that it was a clonal acquired disease, but it required the actual identification of the gene, which is a gene called PIG-A that I've shown you above, which become mutated at many different sites. It's an X chromosome gene, but the disease occurs in both men and women. But it means that a mutation in just one copy is sufficient to change the phenotype, the behavior of the red blood cell, so that it becomes very sensitive to complement. And complement is part of the innate immune system. It's activated all the time, perhaps more at night. Maybe it's the explanation for the occurrence of the hemolysis in the morning. But explaining how a single gene could lead to the disappearance of upwards of 50 different types of proteins on the cell surface was quite mysterious until the gene itself was discovered and characterized, and it was shown to be in some enzymatic fashion responsible for an early stage in the synthesis of what's shown on the right, the glycosyl phosphatidyl inositol anchor. And this is a complex chemical moiety that anchors these proteins to the cell surface. So a deficiency in the synthesis of this anchor leads to the absence of a whole group of GPI-anchored proteins of diverse function on the cell surface, some of which help to inactivate the complement that's constantly being renewed in the bloodstream and would ordinarily lyse red cells if there weren't protective membrane proteins. Now, I told you that PNH is common in aplastic anemia, so I've been familiar with it for my entire career, and upwards of 50% of patients with aplastic anemia and some other similar hematologic diseases will show PNH cells in their blood, even if it doesn't cause this full syndrome that I've just described to you. So there's a very intimate relationship that was recognized even in the mid-20th century, just on clinical grounds before we understood very much about either disease. So it's this background that has led us to speculate that clones, like the PNH clones, may arise under very specific circumstances. This is just a schema to give you the framing of how somatic mutations appear in hematology. I've shown you in the middle the somatic mutations occurring in the setting of the disease, immune attack on the bone marrow, but in the setting of cytotoxic therapy, as shown at the upper left, there are other clones that are truly pre-malignant, for example, are mutated in TP53, which are selected in the setting of that type of stress on the bone marrow. And then the CHIP, or clonal hematopoiesis of indeterminate prognosis, or age-related clonal hematopoiesis, shown at the lower right, occurs with normal aging, perhaps as a result of exposure to chronic inflammation, to stem cell depletion, other diseases, and to toxins. So this is kind of Darwinian in the sense that we've got an environment that's acting to promote clonal expansion. Now, VEXAS, you're going to hear about from Peter, but it's a completely different disease, although again, one that's very diverse in its manifestations, as Peter will emphasize, mainly rheumatologic, but although I'm a happy collaborator with my rheumatology colleagues, we're going to claim this as a hematologic disease because it arises in an hematopoietic stem cell. This is just one example of the hematology of VEXAS. You'll hear all about the vasculitis and the collagen and joint problems that occur in VEXAS, but VEXAS also causes real bone marrow problems and even changes the appearance of the bone marrow. And myelodysplastic syndrome is a syndrome of marrow failure as well as a pre-malignant syndrome, and that's quite common in our patients with VEXAS. So again, like PNH, a multi-system, multi-organ disease with very serious manifestations for me as a hematologist in the bone marrow. Now, Satu and I took great pride in this paper, and I hope people in the audience read the whole thing, but at the least, please do look at the figures. We were very, very proud, and we've got a terrific Nordic graphic artist to try to get our ideas into a vivid manifestation as pictures. And I'm going to walk you through the figures in more detail and try to emphasize what we took from the bridging between this early syndrome of PNH and VEXAS, discovered just a couple of years ago by very sophisticated genomic approaches. And our realization, we didn't discover anything here, but our realization was that these syndromes had really been hiding in plain sight, that different medical specialties, pediatrics versus medicine, pathologists versus internists, had understood that some diseases occurred, non-malignant diseases occurred as a result of mutations, but no one had really put them all together and speculated, and I think make a good case that these are very common mechanisms of disease that are only now beginning to be appreciated with the application of single cell and next generation sequencing techniques to a whole variety of diseases. So the first figure really had to do with mutations, and mutations turn out to be really complicated and not at all straightforward. And we made a sort of jocular comment in the paper that the New England Journal editor allowed us to keep, that we think of mutations in a bad way, mutants, the X-Men, that they're terrible and also relatively rare, that they stand out as being unusual. And the actual reality is quite different. First of all, they're not rare at all. And although the rate at any particular nucleotide is really low with each mitosis, as Dan would tell you, the genome is really big. So when you've got a very large scale in terms of the number of nucleotides that can be worked on, even if the rate at each nucleotide per cell division is low, the cell ends up accumulating hundreds to thousands of mutations just in the course of normal fetal childhood development and over the course of a normal life. And I think that most medical people, including me, had a sort of tabula rasa feeling about cells. They were all fine, they were normal, and it was only the occasional mutation that led to the catastrophic outcome of a cancer. But the reality is that every cell is full of mutations. Now, the good thing is that most of the time, these mutations have no functional effect, or they just kill the cell and the body doesn't need to worry about it. But in every organ, the bulk of cells have accumulated large numbers of mutations, not all of which are going to be harmless or neutral. Now, we know from cancer biology that cancer somatic mutations, and there's every reason to believe that this would be true of all somatic mutations, really arise mainly as a result of replication errors. The more time a cell has to undergo a mitosis, the more likely a mutation is to arise as a result of inadequate repair of the inevitable errors that occur with replication. And in fact, Bert Vogelstein and Tomasetti up at Hopkins showed quite nicely a theoretical paper that the likelihood of a cancer in any organ is basically a function of the number of mitoses between the stem cell in that organ and the mature cell. Most of the mutations that we see that occur as a result of aging have a very specific signature of genetic abnormalities. And those are due to the cytosine, methylcytosine deamination events, which occur partly as a result of errors during replication, but also occur simply as a result of removal of the amino group from a cytosine, which creates essentially a uracil that mispairs at the next mitosis. And that event of deamination probably is strongly related to our exposure to reactive oxygen species and to inflammation. And this is simply inevitable and age-related. Now, recent data have shown that mutations occur not only abundantly in any cell, not only as a result of inevitable mitosis or inevitable exposure to reactive oxygen and nitrogen species, but that mutations occur very early in our lives, early enough to be categorized as post-zygotic. There's the sperm and the egg come together, there's a zygote, and mutations start to accumulate at this very early stage. And in really extraordinary computational mapping of mutations that cause disease, including benign disease, late in life, the original mutation can be traced back in many patients to these early stages of embryonic development. So at the very beginning, we think about the fetus being replications are occurring, mutations are occurring. Most of them are going to be harmless, but harmful mutations can occur then and not become manifest as disease until decades later. And when we model, again, I'm not going to go through the details of this also remarkable paper of a few years ago. When you take large data sets and you look at the clonal hematopoiesis, these clones that I mentioned to you like CHIP, age-related mutations that lead to clones of cells that carry a mutation in a specific cell, they're not occurring as a result of neutral drift or randomness. The mutations are occurring randomly, but the clones are growing almost certainly because they convey to the cell an advantage in the sense that, for example, the cell's ability to self-renew is increased and its desire to undergo differentiation is decreased, or the cell doesn't undergo programmed cell death or is resistant to environmental factors that might ordinarily kill it. So the clones that we see represent selection in the environment. So we have the ingredients of what I'm going to stress as my talk ends of heterogeneity and the genetic makeup of cells within organs and an environment that is going to select for certain clones due to their features. Now, this is the illustration that Satu and I in this very complex area, thought best showed some of these events that I've just described. Every tissue is genetically mosaic, and we tried to show that with colors and with the sizes of clones. Sometimes it's unlikely, it's just a single cell that carries a set of mutations, but in some instances that cell has a proliferative or other advantage and becomes a clone, meaning that there are multiple cells that now carry a mutational load. And as I mentioned, while most of these somatic mutations are neutral, that's probably the case for all those very small circles. Deleterious mutations mean the cell's eliminated. We don't see any other cells. But in other instances, the clones are positively selected. And as I've shown on the left, some of these mutations occur quite early in life. Some of them, as shown on the right, can go on to cancer, but there are other clones that proliferate, expand, but have functional effects on the progeny and change the way the organ, or in fact, the organism behaves. Now, Darwin really was extraordinary. And going back and reading Darwin, going back and reading The Origin of Species is illuminating, I think, for any biologist. Obzansky made this famous comment that everything in biology has to do with evolution. What's remarkable in The Origin of Species is that Darwin only had one figure. It's quite a contrast to our PowerPoint talks. This is the figure that I'm showing you here to show that natural selection worked to produce organisms in our own time period, shown at the top, with extinction and a change, which he called descent with modification. He actually didn't use the term evolution very much. And these comments of Darwin, I think, are, written in this wonderful early 19th century English, are still, I think, quite impressive as we read. Now, of course, Darwin's talking about the struggle for life among organisms. I'm going to point out some of the similarities, but also some of the differences between evolution as it occurs among organisms and evolution that occurs in tissues. Now, Darwin, he was really important, really influential, really insightful, but he had a different perspective from us. And when you read the Origin of Species, his knowledge of life, not just the life of us simians, but of all organisms, plants, pets, the shells, mollusks, is extraordinary. And of course, his emphasis had to be on the attention to the details, these small differences among animals. Sexual transmission was something that he struggled with. How can traits actually be transmitted? And he had lots of issues that he had to confront the people he was fighting with that are perhaps of less relevance to us. He also had, unfortunately, no mechanism. Genetics had yet to be really discovered, so he didn't really understand the basis of what he observed in evolution. He wasn't emphasizing the medical aspects. He had no pathophysiology, and he really dealt more with things like the role of life forces and the role of a creator, rather than what are now very familiar to evolutionary biologists, as, for example, statistics and population genetics. I want you to read for yourself these comments, because I think they still really apply to us. And one of the things that Satu and I struggled with was the scale, the number of cells, the number of mutations, the length of life when it's perceived in nanoseconds. And I think it's really very hard sometimes for us, even if we're scientifically trained, to get our heads around the scale issue. The credit also has to be pointed out for August Weismann, who understood the difference between what we're talking about today, which is the evolution of an organism, evolution of a species, the origin of species, which occurs through the germline, which involves sexual selection and heredity, and what occurs in the soma. And he understood that there was this barrier between soma and germplasm as part of the explanation for how heredity might work, even prior to genetics. But of course, the emphasis in the lectures today is really on what's occurring in the soma, what is not inherited and what is not heritable. Now, there are big differences that I've just summarized here between Darwinian evolution as it applies to the germline and the soma. Organism versus cell, mutation rate, which is much higher in somatic tissues, at least tenfold higher, because there's less preoccupation with the fidelity of transmission of genetic information. Selection among organisms in life is mainly negative. It's getting rid of the less efficient, the less favored members of species, where somatic mutation appears to be almost entirely positive. something about the cell that allows it to survive and thrive. An increase in the mutation rate may be beneficial for germlines because long-term it leads to changes that lead to new organisms, more successful organisms. It's almost always harmful in somatic tissues. And there are other differences in terms of historic drivers, the role of the environment, how traits are actually provided. So I want to spell out a few of these things, flesh them out in just a couple of slides. Before I get to that, I want to emphasize that Darwin, in the sense of heterogeneity that has to be acted on by an environment, is fundamental. That's the simplest description. Descent with modification as a result of environmental factors is the simplest statement that you can make about Darwin. But Darwin doesn't turn out to be simple in any way. I won't go through this at any depth. I'll give you a couple of examples, but evolution is really complicated. From what exactly is being transmitted? Is it the DNA? Is it an instruction in terms of genetic proteins? The role of plasticity, the evolvability of traits that may make them more heritable or more resistant, and of course the role of the environment in altering organisms, and also the role of the organisms in altering their environment. So a couple of examples of some of the similarities or analogies, and I hope provocative ones, between classic Darwinian evolution, some of the neo-Darwinian synthesis, and what's going on in somatic tissues. So one is that Darwin really struggled. I colored in his single graphic in the origin. Darwin really struggled with the whole question of what is a species. They're intermediate forms, they're hard to find. What makes a species different? How do we actually define a species being different? Actually, I think evolutionary biologists are still struggling with how we actually define a species, or whether a species even is a real entity. And that's really not different from what we deal with in somatic mutations. For example, there are plenty of cells in every tissue that have a full panoply of cancer gene mutations, and they never become cancer. So the distinction between a true cancer cell and a cell that has those same mutations is not so obvious when we do deep sequencing of single cells or of clones of cells. Really a struggle to define a cancer cell, or to even know in benign diseases when the phenotype is altered sufficiently to make it distinct. A second extension from Darwin via other evolutionary biologists, and I'm especially grateful to S.J. Gould's work for this, is how do we view evolution? And I think that the sort of common sense view, maybe the hopeful view of evolution, is shown at the upper right. That we've got evolution to an end, and a nice end. I mean, you've got smart, beautiful, whatever people there, and that the idea also, as Darwin spelled it out, is that we're getting to the species that we see around us, and there's a progression. We all remember this from our biology courses of the evolution of the elephant, the evolution of the horse. They all get to be like what we think of as the ideal, which is the elephant we have now and the horse we have now. But evolution doesn't have a goal. It just does its thing, and whatever the environment selects is the end point of evolution. It's not going anywhere, it just goes. In Gould's wonderful book, Wonderful Life, he describes multitude of species that are completely different from anything that exists today and just got wiped out. They're gone, and there's no intrinsic reason for us to think that they were better or worse, or wouldn't deserve a nicer fate than extinction, but evolution and the environment do what they're going to do. And I think there may be some human parallels as well. It's nice to think that we're all heading towards an Einstein, but out of that enormous variety, what really succeeds may not be what we would ideally like to believe. And the third and last thing I'll say just about this parallel is that evolution remains complicated. There's a famous paradox in classic evolutionary biology as to why, this is after Peto, the statistician, an English statistician, why do large animals not all die of cancer? Because we got so many more cells, and if it's simply a a stochastic risk that you acquire the mutations for cancer, then why do big animals not all have cancer? Because they have so many more cells, period. Now, there are some partial answers in terms of telomere biology or multiple versions of TP53 that might provide an answer, but it's a paradox that was worth looking at. And we have paradoxes as well in somatic mutations. So, for example, in hematology, we know that mutations occur in all the lineages of the hematopoietic stem cell, the cell that's going to give rise to red cells, white cells, and platelets. But it's really only in the only occasionally that we see malignancies in some of these lineages where they're much more typical in others. So chronic myeloid leukemia developing into acute myeloid leukemia is quite typical of what happens in white cells of unfortunate individuals who develop leukemia. But we have mutations that occur in the precursors of red cells and of platelets, as well as in PNH, that very infrequently transform to leukemia. And we don't really understand why mutations in one setting, even in intracellular or in a tissue environment, may have one consequence versus another. So it's complicated. And in our Darwin paper, in our paper in the New England Journal, Satu tried to show the multidimensionality. Of course, we're stuck on this, on a two-dimensional surface, by showing that the depth at which the variability can occur is really quite extraordinary. And even looking at something as simple as time, time may be chronologic, but there's also the aging time, inflammaging, or whether time really passes the same when you're young and old. I can tell you that it doesn't from my own experience, that mutations that occur have to interact with other mutations. They affect multiple different types of cell function. And of course, there are all these interactions with environment, both intracellular, the local environment, and the bigger environment around us. So finishing up and talking about the actual diseases, well, there are a lot of them. So here's our summary of all the different organs at the time of this review in which somatic mutations can clearly be related to disease. I've already mentioned CHIP, in which we think that the major consequence of clonal hematopoiesis of indeterminate prognosis is not at all in the blood system, but may have to do with atherosclerotic disease and pro-inflammatory macrophages and myelocytes. You're going to hear about VEXAS. There are other syndromes, myelodysplastic syndrome, myeloproliferative syndromes, in which the functional effects of mutations are quite obvious. Here's a list of all those, the genes, all of the somatic mutation diseases that Satu could come up with. And I want to just go through some of the distinct types. I've mentioned hematopoietic hematopoietic cell mutations, but there are also topographic mutations that lead to nevi, to vascular malformations, probably the cause of uterine dysfunction that's considered benign. I've told you about the hematopoietic cells. I'm going to tell you a bit about immune response and also immune system abnormalities. So here are the topographic mutations that I referred to. These are well characterized, very clear mutations that result in this. And for example, endometriosis has been now well described as a disease in which a benign mutation, probably occurring very early on in life, leads to this painful gynecologic ailment. Here are the stem cell mutations that I just told you about. Here are some mutations that occur in the immune system, and again have been recognized previously as being sort of peculiarities, that we have genetic syndromes, Dan and Peter are experts in these, that are inherited, but also may arise as a result of a somatic mutation in the same gene. Loss of function in immunity may be leading to immunodeficiency or dysregulation, or gain of function leading to hyperstimulation of one component of the immune system. Here are some examples of gain or loss function mutations. Large granular lymphocytosis, pure red cell aplasia, aplastic anemia have now been shown in some instances to have at least a contributor and maybe an etiology in mutations that occur in the STAT2, STAT3 genes, leading to increased signaling that's not properly regulated in a very small subset of lymphocytes and perhaps also in macrophages. And finally, mutations that occur can also be a stimulus to the immune system. They can present neoantigens or aberrant or increased antigens in target tissues that lead to a normal immune response. We think that these are responsible for some forms of bone marrow failure, but they also seem to me to be highly likely to be responsible for a whole variety of autoimmune diseases. So we now can think about autoimmune diseases, so-called benign diseases with really quite awful consequences, may be arising because of mutations in lymphocytes or mutations that have effects on mature macrophages arising in hematopoietic stem cell, or mutations that occur in a tissue that lead to appropriate immune responses to an aberrant antigen. That's everything I wanted to say. I hope I didn't go too far over my time, and I'll take questions at the end. Thank you again, Dan and Wynn, for the invitation.
All right. Well, Peter, would you like to take over at this point? Neal, that was really a wonderful presentation, and we'll have, I'm sure, lots of questions at the end. But for now, we'll turn things over to Dr. Grayson, who's actually going to have an interview with the patient as part of his presentation. So, Dr. Grayson, please take it away.
Thanks, Dr. Kastner. You can see my slides?
Indeed.
Great. Well, first, I'd like to thank Dr. Young for inviting me to co-present with him in that wonderful talk. And thanks to Dr. Kastner for moderating and Dr. Arias for implementing this really special lecture series, of which I am honored to be a part. And also thanks to Mr. Randy Fersen, who you're going to hear from at the end of the talk when we conduct a patient interview. I have no disclosures, and I must say my one disclosure is that this feels a bit like a family affair, because I have been working closely with Dr. Young's group and Dr. Kastner's group for a lot of the data that I'm going to show, and I can explicitly testify that I consider each of them a mensch. So here are my learning objectives. I really want you to recognize that somatic mutations play a causal role in a spectrum of rheumatologic and inflammatory diseases. And what I get to do is provide some clinical flesh to the concepts that Dr. Young introduced you to. And I'm going to pay particular attention on the pathogenetic mechanisms of and the clinical features of the VEXAS syndrome. My hope is that from this talk, you'll be able to understand what causes VEXAS, and you'll be able to spot it clinically. I'm going to do something a little bit radical in this talk, and I'm going to start with not one, not two, not three, but 4 complex clinical cases that I'm going to present to you in rapid fire. And I'm going to ask that you hold these cases in your mind independently and think about ways in which they are common, because each case is on the surface going to seem very different. And these are cases that actually could commonly be encountered in routine clinical practice, particularly among rheumatologists. So case number 1. This is a 45-year-old woman who presented with dry eyes, dry mouth, and salivary gland enlargement. She was ANA positive with anti-SSA antibodies. Her complement levels were reduced, rheumatoid factor was present, and a lip biopsy confirmed a diagnosis of Sjogren's syndrome. 3 years into the course of disease, she developed a severe purpuric rash on the lower extremities, shown here. Cryoglobulins were detected, and a biopsy was consistent with leukocytoclastic vasculitis. 5 years into the course of disease, she developed night sweats and fever and was ultimately diagnosed with non-Hodgkin's lymphoma. Case 2. This is a 59-year-old man with ANCA-associated vasculitis who presented to the emergency department with crushing substernal chest pain. His vasculitis was in established remission for 2 years off medication. He regularly visited his primary care physician. His blood pressure and cholesterol levels were always at goal. He never smoked cigarettes. He exercised regularly. He had no family history of early coronary artery disease, and yet a rapid evaluation confirmed an acute coronary syndrome. And he asked, how could this have happened to me? Case 3. An 18-year-old man presents with gradual onset bloody diarrhea associated with severe cramps. He describes frequent bowel movements of small volume. He was diagnosed with ulcerative colitis, but he was non-compliant with his care and experienced several episodes of severe colitis over the next 8 years. Eventually, he returned to established care during a period when he felt relatively well. But surveillance colonoscopy unfortunately detected a mass-like lesion in the sigmoid colon, which was subsequently confirmed to be colorectal cancer. And finally, case 4, which I'll go into in a little bit more detail than the others. This is a middle-aged man who was previously well until 2011, when he developed a bizarre constellation of severe systemic inflammatory features, which included fevers, orchitis, painful skin nodules, periorbital face swelling, recurrent deep vein thrombosis, and a few episodes of swollen, painful ear consistent with ear chondritis. His inflammation was difficult to control with anything other than moderate to high doses of daily glucocorticoids. Steroid-sparing therapies were not effective. And finally, in 2013, he was admitted to the NIH Undiagnosed Disease Program. And under careful observation here at the clinical center, we tapered his glucocorticoid dose from 25 milligrams of prednisone daily to just down to 20 milligrams prednisone daily. And with that slight taper, as he predicted would happen, he experienced a flare in his disease symptoms, which we were then able to characterize. What we saw was that his acute phase reactants, his inflammation markers in blood, went very high, including C-reactive protein levels over 300 milligrams per liter. The periorbital face swelling that he described had returned, which the picture is showing here. And he developed those painful skin nodules that he described to us, and a biopsy showed a medium vessel vasculitis down deep in the dermis. Because he had medium vessel vasculitis of skin, combined with this history of testicular inflammation, we thought the closest approximation of his diagnosis was perhaps an atypical form of polyarteritis nodosa, which is a form of medium vessel vasculitis. And under that paradigm, we tried to treat him for the next few years, and we were largely unsuccessful. He still required high doses of glucocorticoids. At one point, we tried anakinra, which was notable for a severe skin reaction shown in the picture. And in the background of all of this inflammatory symptoms, he started to develop progressive bone marrow failure, which began first as a macrocytic anemia, followed by thrombocytopenia. He continued to have inflammation. He started to develop multiple infections. And in 2017, he underwent a bone marrow biopsy due to worsening anemia, and now he was requiring blood transfusions. His bone marrow biopsy was non-diagnostic. He had hypercellular marrow with myeloid predominance. He had negative cytogenetic and genomic studies for myelodysplastic syndrome, which was the clinical concern. And we just didn't know what this gentleman had. He had this weird constellation of systemic inflammation that was difficult to treat, coupled with progressive bone marrow failure. We soldiered on, and the patient got worse, unfortunately. He began to have fevers, pulmonary infiltrates that were only responsive to high doses of intravenous glucocorticoids. He developed a disseminated atypical mycobacterial infection. In addition to blood transfusions, he now required platelet transfusions for progressive bone marrow failure. He had multiple hospitalizations, ongoing inflammation, and unfortunately in 2020, he died from complications of his disease. And although we didn't know it for the first 9 years or so of his disease, by the last year, we knew this patient to have this newly discovered disease known as the VEXAS syndrome. So these are the 4 cases, and hopefully you can keep them in mind. So what do these cases have in common? Well, there's a couple commonalities among them. For one, in many instances, there was vascular disease. So the Sjogren's syndrome patient who develops cryoglobulinemic vasculitis, the ANCA-associated vasculitis patient who developed an acute coronary syndrome, the VEXAS syndrome patient who develops vasculitis down deep in the dermis of the skin. All the cases had life-threatening complications, and many of them were hallmarked by persistent inflammation that was difficult to control. And finally, there was this theme of evolution to malignancy in some of the cases. The Sjogren's syndrome patient who evolved to develop non-Hodgkin's lymphoma, the ulcerative colitis patient who was non-compliant and eventually ended up with colorectal cancer, and the VEXAS syndrome patient who had bone marrow biopsies done with concerns for progressive myeloid neoplasia or pre-malignant conditions like myelodysplastic syndrome. So I'm going to walk you through the last case first, the VEXAS syndrome case, and then we'll return to the other 3 because each of these 4 cases is actually linked by a common mechanism, and it's the point of the conference today. In each of these cases, a somatic mutation is playing a causal role. So we've alluded to it before, but let me go into more detail of describing what the VEXAS syndrome is. And this is, as Dr. Young pointed out, really an overlap disease that has inflammation features and has hematologic features. It's almost a disease exclusively found in men, which I'll explain in a minute, and in older men, which is why the patient here looks this way. And it's characterized by inflammation in the eyes, in the skin, in the cartilage of the ears and the nose, as well as a significant component of inflammation in the lungs with serositis, pericardial effusions, pleural effusions. There's a tremendous variability in skin involvement. You see a lot of different rashes with these patients. And then hematologic manifestations. They can get recurrent deep vein thrombosis. They can get progressive bone marrow failure, and then ultimately evolve into myelodysplastic syndromes or even multiple myeloma. And to understand the mechanism of this disease and how it works, I want to tell you the story of how the disease was discovered, because this story starts in Dan Kastner's lab with a clinical fellow who's now faculty at NYU named David Beck. And David Beck took advantage of the fact that most patients that come through the NIH Clinical Center have genetic sequencing performed. And in the undiagnosed disease program at the NIH, and as well in Dr. Kastner's periodic fever team clinic, they had accumulated a database of exomes from 2,560 individuals. And David Beck took a pretty radical approach. He said, I'm going to start looking at this genetic data without paying much attention up front to the clinical phenotype of these patients. And I'm going to see if I can make discoveries based on genetic commonalities. And to do this, he restricted his search to 841 genes related to the ubiquitin pathway. He was interested in the ubiquitin pathway because genetic perturbations of this pathway had been associated with a variety of autoinflammatory diseases, which is a major interest of the Kastner group. And he applied some filters to ensure that any genetic discoveries were likely to be pathogenic, including that genes had to be intolerant to haploinsufficiency, novel variants not found in human databases, and he was looking for shared variants across cases. And when he applied this filter strategy, out pops 3 patients who have a missense mutation in codon 41 of a gene known as UBA1. This is the master gene of all cellular ubiquination. And here are chromatograms from these first 3 patients. And to interpret these chromatograms, it's first important to understand that these patients were all men. All 3 of them were male. And the UBA1 gene is on the X chromosome. So in these chromatograms, you see these nucleotide peaks. And because men only have 1 X allele, you should see a single peak at every locus. But what you see is each of these patients have a double peak. And the double peak, you see a reference read, a normal read, as well as a mutant read. And after some housekeeping efforts, it became pretty clear to us that this is likely reflective of mosaicism, meaning that some populations of blood in these patients had this mutation in UBA1, and other populations of cells in blood didn't possess the mutation. Hence, we were seeing 2 peaks when there should have been 1. To confirm this, we sequenced skin fibroblasts from these patients. And here you see single peaks, codon 41, no mutation present. So at this point, we realized we were dealing with a somatic mutation that was restricted to blood. And moreover, it was only found in certain cell populations in blood. So with help of hematopathology colleagues, Dr. Beck actually sorted out different stem cell populations in marrow, as well as mature circulating cells in peripheral blood. And what I'm showing you here are chromatograms from a single patient. The red peak is the normal peak in codon 41 of UBA1, and the blue is the mutation. And if you look at the level of the stem cells, you see an equal red and blue peak, meaning that some stem cells in this patient have this mutation in UBA1 and others don't. And when you chase that through myeloid lineages and end up in mature circulating neutrophils and monocytes, you see a very big blue peak, big mutant peak, and very little normal reads, meaning that most of the cells in circulation in this patient, at least from the neutrophil and monocyte perspective, were mutant. But when you look over here through the lymphoid progenitors into mature T and B cells, you see the normal reference peak, no mutation. So what's happening in this disease is there's a mutation arising in stem cells that's becoming lineage restricted only to myeloid cells and being negatively selected at the level of the marrow in lymphocytes. And shown a different way, we can quantify the size of this mutant clone by a metric known as the variant allele fraction. And here you can see skin fibroblasts, no mutation, mature T cells and B cells, no mutation. But looking in these myeloid populations, there's a really heavy mutational burden. UBA1 is a master enzyme of ubiquination. And what we found through collaboration with Akeem Warner is that this leads to defects or reduction in cytoplasmic ubiquination in these patients and accumulation likely of junk protein. And so when we looked at the bone marrow aspirates in collaboration with Kathy Calvo here from the Clinical Center, we saw these little white dots in the cytoplasm of myeloid and erythroid precursor cells in every patient that we initially first diagnosed with this VEXAS syndrome. So this is a finding that's really sensitive in VEXAS. It's not perfect, not every patient has it, but most do. And we worked with Keith Sikora from NIAMS to start to try to understand, well, what is this defect in UBA1 doing to these myeloid cells? Well, it turns out it's a pretty toxic mutation. This is a bone marrow-derived monocyte from a healthy subject. This is from a subject with VEXAS syndrome. And when we do bulk RNA sequencing of myeloid versus lymphoid populations, we see upregulation of many pathways of innate immunity in the myeloid cells in patients with this disease, including very high levels of IL-1, IL-6, TNF, type 1 and type 2 interferons, which is why in the case I showed you, the disease is often very treatment refractory because targeting one cytokine pathway usually is insufficient in this disease. And VEXAS is an acronym. So it stands for, the V is for vacuoles, these characteristic vacuoles on bone marrow aspirates. E1 enzyme is the enzyme encoded for by UBA1. As I mentioned, it's an X-linked mutation. It produces myeloid-restricted autoinflammation. And the point of today's conference, it's a somatic mutation. It's a mutation that arises sometime later in life. And that makes sense when you start to look at the demographics of who gets VEXAS syndrome. And the one striking thing to really see is this is a disease of late adulthood. The earliest patient we had, the youngest patient, that first symptom was 45 years of age. So this disease presents in the 5th decade of life or later. 100% of the initial subjects described were male because it functions like an X-linked recessive disease. And what was very fascinating to rheumatologists and to hematologists is this syndrome was hiding out amongst a myriad of different clinical diagnoses. So these patients often met diagnostic criteria or classification criteria for a range of diseases, including relapsing polychondritis due to the cartilage inflammation of the ear and the nose, but also diseases like Sweet syndrome, myelodysplastic syndrome, multiple myeloma, polyarteritis nodosa, like the case I first showed you, and giant cell arteritis. And as a rheumatologist, this began to really challenge my thinking about genetics of our diseases. For rheumatologic conditions, like for many conditions in internal medicine, we're starting to understand the genetic landscape of diseases, often through studies like genome-wide association studies or GWAS studies, where we're looking at common genetic polymorphisms in the germline in disease versus a comparator population. And these studies have been really effective at teaching us about potential causal pathways and therapeutic targets in rheumatologic diseases. But when you think about them with the known epidemiology of many rheumatologic diseases, germline mutations don't make a lot of sense. For example, a disease like giant cell arteritis is a rheumatologic condition that's exclusively restricted to late adulthood, 50 years or older. And if you think about it, it's probably pretty unlikely that mutations that you inherited from your parents that are in your germline would cause a disease that starts when you're 85 years of age. But in contrast, somatic mutations, mutations that arise later in life, could really be a causal contributor to adult onset diseases. It also explains the lack of heritability that we see in most rheumatologic conditions. We don't see familial aggregation of cases. In vasculitis, for example, it's exceedingly rare that we see more than one family member with the disease, which really argues against Mendelian genetics. It argues against germline genetic contributions. So now that we had found this VEXAS syndrome and we'd realized it was hiding out in rheumatologic diseases, we can go back and start to sequence cohorts of patients and see what percentage of patients with this rheumatologic diagnosis actually has VEXAS. So this work was led by Marcella Ferrata, who is a rheumatologist in my group, who's been studying this disease, relapsing polychondritis. And in our first 92 patients in our cohort here at the NIH, we found 7 patients that actually had mutations in UBA1, meaning that 8% of relapsing polychondritis in our cohort was actually VEXAS syndrome. And Yiming Luo, a fellow in our group, created this really nice, simple algorithm to spot VEXAS syndrome. I think it's really important for clinicians to be aware of. It works with 97% accuracy in our RP cohort. So if you have a patient with ear or nose chondritis, and that patient is male and either has an MCV greater than 100 or platelets less than 200, this model would predict that they would have the VEXAS syndrome with near perfect accuracy. And this is the key thing for rheumatologists and internal medicine doctors to be aware of, this macrocytosis, elevated MCV. When I thought about it in medical school, I thought about it with nutritional deficiencies, alcohol use, or even toxicity from medications that we give in rheumatology. But now I've come to realize if you have a patient with systemic inflammation that's particularly difficult to treat, and that MCV is greater than 100, you really need to consider the VEXAS syndrome. So we learned that the somatic mutations have diagnostic value in adult onset rheumatologic diseases, but it gets even cooler to clinicians and more helpful to clinicians to help take better care of patients, because we're starting to realize that the molecular data also has prognostic value. And 95% of mutations in VEXAS, they're missense mutations where a methionine becomes either a leucine, a valine, or a threonine. And these are survival curves in our cohort of VEXAS syndrome, looking at survival stratified by genotype. And what you see is a genotype-phenotype relationship where patients who have the valine variant are at an increased risk for death relative to patients with the leucine and the threonine variants. And I should also emphasize that these survival curves were made from data before we often knew about this disease. And our goal right now is to discover more effective therapies to shift these curves. And that's starting to happen. Now that we understand causality amongst some of these rheumatologic conditions, which is something we almost never do, we can start thinking about the thing that we all want, which is cure. And within a year of publication of the VEXAS syndrome story in the New England Journal of Medicine, we're starting to see reports of patients with VEXAS syndrome being cured by allogeneic hematopoietic stem cell transplantation. And Bhavisha Patel and Dennis Hickstein here at the Clinical Center are launching a bone marrow transplant protocol to perform transplant in candidates with VEXAS syndrome. And finally, this opens up a whole other range of potentially useful tool to clinicians, because somatic mutations can be quantified. The burden of clone size can be measured and tracked, and it's highly likely that this is going to serve as a biomarker. Can we use this to monitor treatment response? Can we use this for prognostic value? There was a really nice editorial that accompanied the first description of VEXAS syndrome, talking about mutations occurring during embryogenesis, fetal development, infancy, even out into adulthood. And depending on when the mutation occurs, a patient can be more or less mosaic. And here are a whole host of pediatric syndromes where somatic mutations are occurring in early embryogenesis. But I want to focus us in the last bit of my talk out here in adulthood, because this is, as Dr. Young alluded to, an area of expanding research. We're discovering more and more connections between somatic mutations and adult onset diseases beyond just the VEXAS syndrome. What I take away from listening to Dr. Young talk is that the genome is unstable. We are polyclonal individuals. Every cell in our body mutates at least once every week or two. The highest rate is in colon, lowest is in blood. And that intrinsic factors like aging and extrinsic environmental exposure factors contribute to that rate of mutagenesis. And to a rheumatologist, this opens up the possibility that inflammation is both driving and a consequence of somatic mutations. So the VEXAS syndrome is a somatic mutation in stem cells that's producing pro-inflammatory blood. So it's driving, somatic mutations are driving inflammation. But I'm going to show you a couple of examples of the converse thing, where inflammation is actually increasing the rate of somatic mutagenesis. And so with that in mind, I think for rheumatologic diseases, somatic mutations are going to be causal in subsets of patients. They are going to sustain or even eliminate disease at the tissue level in other subsets of patients. And I think they're also contributing to disease-associated events that occur late into the pathogenesis of disease. The best studied tissue compartment for this is blood, because it's easily accessible. And in rheumatology, as we start to think about somatic mutations, some interesting paradigms begin to emerge. And one of those is that somatic mutations in bone marrow stem cells can become lineage restricted either to lymphoid cell lines and produce autoimmune diseases, or to myeloid lineages and produce autoinflammatory diseases. And I think this is going to be an important paradigm to consider conceptually. VEXAS syndrome is a somatic mutation in myeloid cells in UBA1 causing autoinflammation. There are periodic fever syndromes with somatic mutations acquired in the inflammasome. Erdheim-Chester disease is a somatic mutation in histiocytes. And over here on the lymphoid side, you heard of the example from ALPS from Dr. Young, but also Felty syndrome, a subset of rheumatoid arthritis, is associated with STAT3 mutations in CD8 lymphocytes. So let's go back to our cases and see if we can't tie the thread. So our first case, Sjogren's syndrome with cryoglobulinemic vasculitis and lymphoma. To rheumatologists, these clinical associations are well known. Patients with Sjogren's syndrome, particularly the ones that are hypocomplementemic, that have rheumatoid factor, are at risk to develop cryoglobulinemic vasculitis, and those are at particular risk to develop non-Hodgkin's lymphoma. This paper that came out in Cell 2 years ago kind of provides an explanation for these associations and looks at the role of lymphoma driver mutations in the pathogenesis of autoantibodies. So what they did, I'll summarize this paper quickly at a very high level, is they isolated B cells from 4 patients with Sjogren's syndrome, and they were able to reconstruct phylogenetic trees to determine when somatic mutations occurred within those B cell populations. And what they found is that there was an order of events. So these B cell populations first acquire mutations in lymphoma driver genes like CARD11 or TNFA-IP3, and then there's a series of random VDJ mutations. And these were subsets of B cells that were selected because they were all making rheumatoid factor. And what they observed is that as mutations continue to expand within these B cells went from making a benign version of rheumatoid factor to making a pathogenic autoantibody with diminishing protein solubility. In other words, to making cryoglobulins. And this part is where the money is in my mind. So this is a single patient, and you can see this rogue population of B cells making pathogenic rheumatoid factor in 2011. And over 2-year follow-up, the population expands and further mutates. And what happens to this patient in between? This patient develops cryoglobulinemic vasculitis. And so it's probably not a stretch of imagination to envision the scenario where these lymphoma driver gene mutations are producing pathogenic rheumatoid factor, and then that is what also puts patients at risk to develop and evolve into lymphomas. Case number 2, ANCA-associated vasculitis in remission with acute coronary syndrome. We in rheumatology have excellent medications after decades of hard work. The expectation for most rheumatologic diseases is we can put a patient in clinical remission. So what gets these patients in trouble later in life? Or what do patients with rheumatologic diseases commonly die from? And the answer almost always is cardiovascular disease. And when you look at risk factors amongst our patient population, they do not usually have traditional risk factors for accelerated cardiovascular disease. So what is the mechanism? Well, in this patient, I didn't share with you, but when we sequenced his blood, he had a DNMT3A mutation in the clonal population of blood cells at a variant allele fraction of 15%. And so this patient has clonal hematopoiesis of indeterminate potential, or CHIP, which is a concept introduced by Dr. Young. But just as a reminder, there are around 40 to 50 genes associated with myeloid leukemias and myelodysplastic syndromes. And if you sequence bone marrow or peripheral blood from healthy populations, you can detect at an alarming frequency small clonal populations of cells that have mutations in leukemia genes. Now, these patients are at risk to develop future leukemias, but that's not the only problems that they get. And as Dr. Young pointed out, DNMT3A and TET2 or some of the most common CHIP genes, DNMT3A is what our patient had in the case that I showed you. So Dr. Young pointed out that there is this association between CHIP and cardiovascular disease. And what's important to understand is this is a risk that's independent to traditional cardiovascular risk factors at a twofold increased risk in patients that have CHIP versus don't. And in murine models of atherosclerosis, introduction of CHIP mutations can accelerate atherosclerosis because myeloid populations that possess these mutations are pro-inflammatory and produce lots of inflammatory cytokines like IL-6 and IL-1. So it's not that stretch of imagination to think maybe CHIP is one of these mechanisms that is explaining why patients with rheumatologic conditions have accelerated atherosclerosis. So in collaboration with Dr. Young's group, we have started to sequence patients with vasculitis, an inflammatory disease, for CHIP. And what we did is we took 3 forms of vasculitis that span the age spectrum. Takayasu's arteritis, a disease of young, here in their 20s and 30s. ANCA-associated vasculitis, a middle-aged form of vasculitis. And giant cell arteritis, a form of vasculitis in the elderly. This is the prevalence of CHIP in normal healthy populations, and then this is the prevalence across the age spectrum of vasculitis. And what you'll notice is that the curve follows that exponential growth curve that you see with age, but it's shifted upward. So what this means is that in our population, age and inflammation are independently contributing to the likelihood of having CHIP. And what remains to be determined, but what's very exciting to conceptualize, is CHIP a novel mechanism for accelerated atherosclerosis in rheumatologic and other systemic inflammatory diseases? And finally, our last case, the case of a young man with non-compliant who had severe inflammation in the gut and developed colorectal cancer. There have been a lot of studies only in the last couple of years related to somatic mutations in inflammatory bowel disease. This is one of them where they sequenced colonic crypts from 41 donors versus 46 patients with IBD. And you can see here in the controlled healthy population, there is a number of somatic mutations detected in colon tissue. That number and the rate of mutations increases in inflammatory bowel disease. And there is a specific mutational signature, which actually is due to the fact that inflammation is causing selective pressure on the type of mutations that arise. And in this paper, they report mutations in the Th17 pathway, which is a critical immune component of gut immunology. And what they're talking about in this paper is that mutations in these pathways can both sustain inflammation or eliminate inflammation in these patients. And it's probably not that far to then imagine that further mutations in this inflamed gut tissue is probably the predisposing risk factor to develop colorectal cancer and a well-known association with IBD. So in conclusion, somatic mutations are continuously occurring in healthy tissue. Somatic mutations in blood and in solid organs may underlie adult onset rheumatologic diseases. VEXAS is a prototype for this. We have a potential to identify these patients early, especially in blood. We have a potential to cure diseases and to prevent late-stage disease complications like malignancies. And the big take-home message is that genetics can dynamically contribute to inflammatory diseases. We just need to expand our thinking beyond the germline. So I'd like to thank some people in the Vasculitis Translational Research Program, a lot of people on Team VEXAS here at the NIH and beyond. And we are going to transition now to the last part of our presentation. So for this, I'd like for you to join me in welcoming Mr. Randy Fersen, who is a patient with VEXAS syndrome, who I have the pleasure of working with in a research capacity here at the NIH. Mr. Fersen is the CEO of Global Analytica. He's the president of Fersen Associates. He is a career CIA intelligence analyst, the author of many books, one of which we probably will get into. And he teaches about advanced analytic techniques to the intelligence community in the US and across the world. So we are really delighted to have him with us. I'm going to interview him a little bit and so that you get a feel for the symptoms that are commonly encountered in VEXAS syndrome. But his medical history is slightly unique, and it's perhaps a story told in 2 acts. So to set the framework for that, Mr. Fersen has had 2 complex medical illnesses that were diagnostically challenging. His first condition went from 2009 to 2014, where he had unexplained progressive shortness of breath. And he learned a lot, I think, from interacting with the medical community during time, and he's written a lot about that. And then on the heels of that independent medical problem being another set of mysterious symptoms that ultimately ended up being the VEXAS syndrome. So welcome, Mr. Fersen. My first question to you is, can you just briefly walk us through the first of your two complex medical situations, what happened and what your experiences were like with the medical system trying to get diagnostic answers?
Well, thank you, Peter, for inviting me to the presentation and being able to contribute to learning more about these syndromes and VEXAS. Just a really quick round one, which is we're in a 5-year round. I'm a runner and I was having trouble feeling soreness in my lungs when I ran too far or too long. And I was having trouble just with sore lungs. So I went in and they did a major workup at a local hospital and took a look at nuclear stress tests, ultrasound, whatever, looked at my heart. And there's a 3-person team. The junior cardiologist said, maybe he needs a stent. And the 2 senior cardiologists says, no, it's pulmonary. So I was dismissed and I started a metapulmonologist. And then over the next 5 years, I looked at pulmonary function tests, and looking at asthma, looking at allergies, looking at a constricted larynx, looking at acid reflux, a lot more work on allergies. Over 5 years, I'd seen 17 different specialists, including 2 who treated the President of the United States. And I even applied for the NIH Undiagnosed Disease Program. And then I was over in Barcelona. I was really having trouble carrying a 50-pound suitcase around because I was a teacher. And I came back, I was having trouble walking to the State Department. I went in to see my doctor, and she later told me that she'd heard in med school that there's this thing called the look of impending gloom. And she saw it on my face and she said, you're going to the ER right now. So I said, well, I got 2 more appointments and I'll go in this afternoon. She said, no, you're going right now. So I went down to the ER. They gave me an EKG. They looked at me and said, you have no problems. And I said, I'm not leaving. They discharged me. They discharged me a second time. And I said, I'm not leaving. So I pulled out the memo I'd done for the NIH program, just said, take a look at this. And this is what I've gone through for 5 years. And the question was, what do you tell my wife if I die tomorrow? So he says, okay, I give up. I'm going to bring you in. We're going to do a catheterization. That night, they looked, and he came out and he said, I apologize for the cardiac profession, that you're 80 to 90% blocked in every artery. And you're going in for a quadruple heart bypass first thing tomorrow morning. So we did that. 12 days later, I got on a plane and I flew to Montreal, and I actually delivered 3 professional papers at the conference. So that kind of got me through round 1. 2 weeks after that, I started getting this really, really weird rash on my legs, and it was driving me crazy. And that was my second round with undiagnosed diseases. But as having gone through that experience, Dr. Grayson I wrote a book here, How to Get the Right Diagnosis, and 16 tips for navigating the medical system. I learned a lot about going through doctor's offices, because every doctor was a 3 to 6 month round, and actually none of which was actually useful, because they could have been diagnosed correctly the first time and avoided all this. But I learned a lot, and I got a book out, and I think it's going to help a lot of people. And I'll post it if people are interested in the title of it in the chat.
And so on the heels of this 5-year odyssey and then somewhat urgent open heart surgery, the first problem had been resolved. It was occult severe coronary artery disease. And then a new problem quickly began on its heels. And you mentioned that you started to develop rash. Can you tell us a little bit about what that rash was like?
Just what happened is I was having this rash. It was like a very, very deep rash. If you scratch it, it got really intensely painful, and you just had to keep your hands off it. But what really amazed me was that it was on both of my lower legs, and it would migrate around the leg to different parts of the leg. And over a period of 1 or 2 days, it would go from one area, it would move up higher up or move down. So it kept moving on me. And then finally, the left leg stopped hurting, stopped having the rash. And the rash was kind of inside, there was not much redness on the skin itself. So it was just inside the leg was having this rash, which is very curious to me. So we started to figure that out. We went through and we did Again, testing for allergies to hospital products. We did 92 prick tests. We did a wellness doctor looking for parasites. We did diet. We did extensive allergy testing, 26 vials of blood one day, going through all the possible food allergies I could have, any other kind of allergies. We looked at cancer. We did a bone marrow biopsy. We went through that. And all this time, though, I did have fairly high blood pressure, but nobody was paying any attention to that as I was going through the process. So this was around, in essence, I was in an abduction machine is the way I put it, is that they keep trying to eliminate things, and they kept eliminating things. But over 6 years, I was never received a treatment for what was going wrong with me. And I never received a diagnosis or medication from 2014 to 2021. And the word vasculitis never came up. And again, I saw almost 8 different specialties of medicine as I went through this process, and really was getting nowhere.
And one of the things I noted from the chart was that a lot of times when physicians saw the rash, they described it as urticarial or looking like hives, but it didn't feel particularly like the classic hives, right? It wasn't a particularly itchy rash.
Well, I actually, over the course of the 7 years that I was doing this until today, I had different kinds of rashes break out. The one I just described, and one day I had my whole trunk broke out in hives. So they gave me some prednisone then, and it went away in 2 days. I had another very weird rash that came up. But what was really interesting was in June of 21, I was playing with some logs and I got some ant bites. And suddenly I started getting red dots on my arms. And the red dots they're red, they're on the size of a quarter or a dime. And if you pushed them, they really hurt like crazy. But they didn't itch at all. And they really didn't, there were no lesions or anything, it was just the skin. And what would happen is that they would stop hurting after 3 or 4 days, but the redness would stay. So eventually I looked like the monster from the Blue Lagoon, because I had these red dots over my entire trunk, except that I had nothing on my head, but from my shoulders down, I was just covered with these red spots. And we did
severe rashes that were coming and going and responding sometimes to steroids. You started to develop fevers and joint pain as well?
I would have really, like the big knuckle of my left toe suddenly got extraordinarily painful. And for 3 or 4 days, I could barely walk on it. And then it would just disappear. Same thing happened to my right wrist. In fact, they thought, I have a good friend of mine is a hand surgeon. And she says, if it's hurting that much, you probably need to get an x-ray. You probably broke something, from what she could see. And I said, well, let's wait 2 days. Wait 2 days and it stopped hurting. So I had these little spots where in joints or my shoulder. At one point I thought I needed, what's the shoulder injury? The rotator cuff. So they were thinking we're starting by doing rotator cuff surgery. And then suddenly it went away. But that was after a month or 2. So these very strange, also the fevers would come in the middle of the day or at the end of the day, and then they would subside. They always started very low. My temperature went down to the 97s or 96, and then it started to go up, after a couple hours, and I would have chills, and I would have the fever, and then it would stop. It would just happen during the day, and then would be gone. Later on, it started happening more at evenings, but I knew that if I went to bed and when I woke up in the morning, it would be gone. So I couldn't understand the fevers, and I also had, at that point, anemia kicked in. And it went from around a 12 down every week. I went down another point. So I was getting down towards 7 and 8. And then I went and got a colonoscopy, made sure there's no bleeding, and an upper endoscopy. Everything looked absolutely perfect. Fantastic doctor. And then he got me to some other allergists and other people said, there's something really wrong with you. We got to find you other doctors and we got to try to fix this. But that actually turned out to be all negative as well, as they went through very extensive testing of other things that they were looking for.
So you were having skin rashes, then you started getting attacks of arthritis, you're getting fevers, you're feeling progressively unwell, you're getting anemia. And then let's fast forward to an eventful trip that you took to Iceland. And I think you have some pictures to show us from your Iceland trip.
Just give you, I decided it would be fun to go to my 100th country that I've lived in, worked in, or visited. So if I can get, here we go. So here we were doing waterfalls. This was the day before I entered the ER. We're hiking through parts of Iceland, looking at these great waterfalls. I couldn't resist one of my best friends. We stood under the waterfall just to prove that we had truly waterproof clothes on that we bought from REI. And then the next day I woke up and had a fever around, it was 39 degrees, 40. I'm trying to think what, no, it was doing around 105. No, sorry, I'm trying to 103, 104, but I was having trouble standing. So we got in the car, and we went back to the hospital, and we ended up at Landesblad, the level 2 hospital in Reykjavik. And that's where I spent the next 2.5 weeks, getting used to meeting a lot of extremely competent, very capable doctors who were totally focused on diagnosing me. And they worked as teams. They were extremely collaborative. I was most impressed. Everybody in the hospital went on first name, no matter who you were. And of course, I came from CIA, and that's what we did at CIA. So it was the kind of culture I liked, I enjoyed. So let me go back.
And while you were hospitalized, if I could summarize some of the things, a lot of things happened to you then. One was that you had a blood clot.
Yes, there was a blood clot up in my lungs, and also they found one later on down in my lower leg. And that started to complicate my situation. So and was also having some fluid buildup in my lungs. And also in my heart cavity.
So you had effusions and pleural effusions and pericardial effusions that were developing. So fluid around the heart, fluid around the lungs.
And the second day in the ER, I was so exhausted that I was laying flat on the bed. I wanted to drink water. I looked at the glass, and I couldn't raise my arm to pick up the glass. I had to concentrate for 10 minutes until I could get enough focus that I could actually go over and get the glass and take a sip. I mean, basically I could barely move anything in my body. And they said if I hadn't shown up at the hospital and hadn't gotten treatment, that they thought I would have been dead within a month, easily.
And one of the things that they did there is they thought outside the box a little bit because you had unexplained inflammation. Your blood tests were very abnormal. So they performed a temporal artery biopsy. Yeah,
that was a 30-minute operation. They did extremely good job too. But they did that, and then they were looking for a giant cell as a potential hypothesis. I think we looked at, we probably have over 50 diagnoses that had been disproved or considered as we've gone through this process. In fact, we made an argument map of the entire process, and I've shared with Dr. Grayson to say, we've worked through everything over a long period of time, trying to narrow it down to where we ended up.
And the temporal artery biopsy did show some mild inflammation on the outer edges of the artery. It wasn't fully consistent with giant cell arteritis, but it was possibly consistent with that diagnosis. And that's where I actually met you, is that I happened to be calling one of my rheumatology friends and colleagues who I did my medical training with in Boston, who is a rheumatologist from Reykjavik. And I was calling him about a collaborative research project, and he happened to mention your case to me and said that you lived in the DC area. And as he was telling me the case, it caught my ear that he mentioned the anemia and the blood count problems you were having. So in light of the VEXAS syndrome story, I asked him, well, what is your MCV? And he said it was well over 100. And at that point, we eventually, when you returned to the States, we got your blood and we sent it to Dr. Kastner's group. And a little while later, took a little while for the genetic testing, but eventually we were able to confirm a genetic mutation in the UBA1 gene consistent with VEXAS syndrome. And I think from the clinical perspective, a lot of the symptoms that you had really make sense under VEXAS syndrome, that the bad skin inflammation, the blood clots, the fluid around the heart and the lungs, the daily fevers. In the few moments we have left, I wanted to first make a comment that I really did enjoy reading your book. And one of the things that struck me about it was you were advocating very strongly that in complex cases that patients and physicians are a team, that they can actually work together to make a diagnosis, which is so simple an idea that it is radical, because that probably wasn't your experience with many physicians. I wonder if you could expound a little bit about what you mean by ways that physicians can do better in partnering with their patients through these challenging situations.
Yeah, so in putting the book together and kind of the key takeaways that I had, the main one was that the patient has to work in conjunction with the doctor to explore, which also means that the patient has to get the doctor's attention. The doctor's seeing tons and tons and tons of people. You want them to go home and tell their spouse, boy, I had this really interesting case today. But that means you've got to support the doctor and what the doctor's doing. Number one, don't tell the doctor what you have. Tell them what your symptoms are. Let the doctor do the diagnosis and butt out. Don't spend too much time on Google. The biggest problem I've run into in the US system, which I saw none of in Iceland, and I just posted a blog that I did on the 10 best practices that I saw in Iceland medicine, is that you need to get the doctors to talk to each other. And it is extraordinarily difficult to do that. I actually had been seen by a hematologist and by a rheumatologist a month before I went to Iceland, who were trying to figure out what's really going on with me. And I couldn't get them just to call each other before I left for Iceland. So I left for Iceland without having this 2 weeks done, and they were doing their thing. And so when I wrote the book, I was very sympathetic for what the doctors are up against and how hard it is to try to run a system that works. Some of the other things, the need to challenge your assumptions. Nobody ever thought in the medical profession that my initial diagnosis was just wrong. You know, you can say he's having problems. It could be his heart, but they said it wasn't. So every specialist stayed in their box and never thought about challenging what the cardiologist said. Also, listening to people, I found it's really important to find listeners. There, I like the nurses. They're really good listeners usually. And so the nurses are kind of the hero of the entire book for what they've done, and some really good doctors. But you need a collaborative team effort, and you don't want the doctors to be captives of their specialty. They have to, and I had several doctors that just refused to do some. The head of a major asthma and allergy clinic said, I won't tell you, you don't have asthma, what are you going to do? And he says, I'm not going to tell you, it's your problem. And he was one of the leading hospitals in the United States. And he said, this is not what you want as you're a patient. So work with your patient, your partner, make him your doctor, describe symptoms, keep list of your medications, your surgeries. Every time you see a doctor, have that list with you so you don't have to remember it again. And volunteer to maintain databases for your doctor. I did a lot of Excel spreadsheets, and I'm still maintaining lots of data. And my own point, but if I call it the forgotten 5%. If the system treats you, it doesn't diagnose you in most of my instances. So now, obviously, I'm here, and I found the exception to the rule. But they treat, and they say, and they make money doing treatment and visits, and they're not being paid. And people still tell me there's no code for just doing diagnosis, so you can't charge it. So it means it doesn't happen. And if you got to see somebody every 15 minutes, forget it. So if you're one of those 5%, and I do think in a standard deviation, within 2 standard deviations, 2 treatments where the body will cure itself. But if you're down in the 5% tails, you're in big trouble, because the American medical system is not going to expend the energy to try to figure out what's wrong with you, which is why we have NIH, right? So I'll stop it there.
Well, thank you so much for sharing your story with us and putting a face on a new disease syndrome. I really do appreciate it. And I'll turn it back over to Dr. Kastner to moderate the last part of the session.
Well, thank you so much, Drs. Young and Grayson and Mr. Fersen. This was really spectacular, very, very interesting. So I think I will take the moderator's prerogative and just ask another question to Mr. Fersen. So you were in, it sounds like, quite a predicament in terms of your medical situation. Were your doctors able to come up with some sort of a treatment that has helped you? It seems like you're better now than what you're describing from before.
Well, we're right at that stage this week. I'm at 12.5 milligrams of prednisone. I'd like to get it down, and I know that I don't want to get it down fast. I want to get it down really slow. And then the question is, can we come up? Well, I'll let Dr. Grayson maybe jump in here and say, we're looking at a couple other drugs that maybe will allow me to continue to move forward. But it's been since September is when I had the Iceland incident. And so we're almost at well, I'll turn it over to Dr. Grayson.
Okay, so it's been roughly 6 months since the Iceland situation.
Right. Trying to work through, find the information, and see, but the other thing I did leave out, which is actually pretty significant, I think, is that when I had the first 5 years of round 2, the big identifier was very, very high levels of eosinophils. My eosinophil levels were like 10 times normal. I mean, really, and they would be 10, 7, 8 times normal. So that's what they were trying to diagnose, is why do you have these eosinophils? And at one point, I even talked to one of my doctors saying, could they have left something in my chest cavity, that my body's still reacting to, because we run out of hypotheses for how do you explain these eosinophils. So that's what they were focused on, but they never got into the vasculitis arena in that whole 6 years of looking.
Well, I'll turn the Klieg light on to Dr. Grayson to see if he has anything to illuminate us with.
Well, I would just say that Mr. Furson's early in the process. He was diagnosed only a month or so ago. He's responded very well to glucocorticoids, like most patients with this disease do. And we are trying to then think about what medication can we give him to replace the steroids, of which there are increasingly a number of new options. It's been quite amazing to watch investigators around the world start to identify this syndrome, and we are learning from each other internationally. There's been quite rapid advancement in thinking about new treatments for this disease. And so we are in that stage of planning which steroid-sparing therapy to try. With respect to the eosinophilia, I didn't talk about that at all with VEXAS syndrome, because until we met Mr. Furson and one other patient a week or two ago, we didn't realize that that was a feature of this disease, but now we're starting to think that it is and trying to figure out the mechanisms of that. And I think that Dr. Kastner, you probably know this better than anyone, but with discovery of new conditions over time, you watch the clinical phenotype expand and grow and learn new things along the way, which is why partnering with people like Mr. Furson is really great, because we're learning with alongside each other.
Yeah, I was about to ask the question. I don't remember any of the cases that I've seen with VEXAS having had hypereosinophilia, especially as Mr. Furson has described. And I was thinking, well, maybe I just have, I've missed it. I've gotten involved in other things and just don't know about that. So it's reassuring to know that I haven't been, so out to lunch that I missed that along the way. Actually,
it's reassuring to you, but I'm a little worried about me. I don't know how unique I want to be in the world. That's where it comes down.
Well, there's one other patient, at least.
That's right, the two of us.
Yeah, it's always not a good thing when a physician tells you that you're a very unique case. That's for sure. Well, we have some questions coming in from the assembled audience. So one of the group is asking or making the comment, first, amazing science with great potential for new diagnostics. How much of the science has been already translated into making the correct diagnostics of such complex cases? Maybe I'll start with Dr. Grayson, but certainly Dr. Young will have comments on this as well, I'm sure.
Well, I would say here the recent happenings with the VEXAS syndrome are really illustrative of the challenges, because now we have a genetic diagnosis for a disease that is based on a somatic mutation, and for which there aren't a lot of commercial testing opportunities yet. So it's difficult for patients to get diagnosed. So the discovery has been made, and now the diagnostic technology has to catch up. I would say that Lorena Wilson and from Dan Kastner's group in particular has been doing an incredible job of dealing with, over 100 new cases of VEXAS syndrome since we first described the disease a little bit over a year ago. And most of the diagnostics, at least in the early days, have been happening here at the NIH. And the challenges now is trying to make these diagnoses more quickly and make the test more readily available. And that does present challenges to commercial sequencing companies who are used to making germline diagnoses and less used to making diagnoses with somatic mutations. And Dr. Young, I think, can speak to that type of diagnostics, because in the hem-onc world, they're quite familiar.
Yeah, so I mean, the nice thing is that with genomics, you can expand pretty quickly. And the company that we partner with just added UBA1 to the MDS panel. So that's available commercially, and that undoubtedly will be picked up in the private sector and in other academic institutions that aren't familiar with VEXAS, and they'll be alerted from the blood testing laboratory that this is a diagnosis of VEXAS with certainty. And in terms of the larger question of whether you actually translate to treatment, the answer is yes. And PNH is actually a very striking example, because with the understanding of the physiology, the pathophysiology of PNH, there's an extraordinarily effective drug for the hemolytic anemia. It's actually worth a book. It was developed and became the most expensive drug in the world for a long period of time. It is based on the understanding of the absent proteins and their influence on complement, and completely reverses the intravascular hemolysis that occurs by essentially blocking late complement. So it doesn't happen all the time in every case, but the better you understand things, the better you can explain them, the more likely it is that you can benefit the patient. And Mr. Fersen, I have to say, I was really appreciative of your comment about NIH. I don't know if it's entirely true, but at least the people who work here like to think that that is what we provide, but on a too small scale to solve all of our country's health delivery problems.
Yes, that's for sure. Maybe just going back for a moment to a comment that that Dr. Grayson made. You were talking about the numbers of cases of VEXAS or suspected VEXAS that you're aware of. Do you have any sense? Can you give us a sense of how common VEXAS is?
Yeah, I don't think we have exact prevalence yet. I've heard through David Beck about some possible estimates, and we have been discussing this rigorously. And the thought has been maybe the prevalence of this disease could be something in the order of 1 in 10,000, 1 in 20,000, which is rather extraordinary for a newly discovered disease. I think because VEXAS is driven by a kind of a novel pathogenetic mechanism, it produces a phenotype that's so complex. These patients are just being dispersed through a number of different specialties. So they see hematology, they see rheumatology, they see dermatology, and the challenges that Mr. Fersen alluded to of communication between those specialties. And one of the challenges in communication is that we don't all speak the same language. You know, Dr. Young had to teach us about the language of somatic mutations before we could even have a conversation in this arena. But as we get that common framework, I think we're going to find these patients and find the disease to be more common than we initially thought, because it's rather easy to recognize once you know the clinical features to look for.
Yeah, I think that you're making a very good point, Dr. Grayson, and the bridge building metaphor that we were discussing at the very outset of this session between basic scientists and clinicians. It's equally important to build bridges amongst individuals of different subspecialties and specialties of medicine and different areas, nurses and physicians and other health professionals, so that we can talk about these kinds of things, because a lot of times it is just a matter of going out and talking about these illnesses to medical groups, and then at the end of the talk, somebody comes up to you and says, "Oh, I have a patient that I think has this," or you get an email the next day about that, and it really is remarkable how often that kind of thing can happen. Well, we have another question that came in for Dr. Young. How do you distinguish acquired germline mutation from a somatic mutation in VEXAS? Does variant allele fraction help?
Well, I'm going to leave that to Peter, I think, or you, Dan, can answer that as easily.
Yeah. So, Peter, do you want to comment on that?
I would love for you to comment on it.
Yeah. Well, so certainly, if someone has an acquired germline mutation, then one would not have, presumably, if it's germline, one wouldn't have the heterozygous appearance that one has with the somatic mutation. And that was initially when David Beck first noticed this, and there was the whole question as to what was going on there. You know, given the fact that it's an X chromosome gene, you know, one of the first things that we thought of was it a sequencing error? Mm-hmm. And in fact, the software that have been designed for analysis of this kind of genomic sequence, by default, throw these things out as being a sequencing error. And it was to David Beck's credit that he looked again and said, well, gee, you know, maybe I ought to think about whether there's really something there. And then he considered the possibility of aneuploidy, of there being an extra copy of the X chromosome, and ruled that out. And then started thinking about what you were talking about, Dr. Grayson, of that there could be separate subpopulations of cells, one of which has the mutation and the other of which does not.
Yeah, that's, I think, you know, the question has a somewhat simple answer in that germline mutations should be seen in every tissue that you examine, and somatic mutations generally are restricted to some tissue. Again, the nice picture that you showed, Peter, that depending on when the mutation arises, its distribution and its frequency in different tissues will be different, but germline mutations are present everywhere. So classically in hematology, when we're debating acquired versus a germline mutation, and that's the distinction, we would look at the skin, or we look at lymphocytes versus granulocytes. But a skin biopsy should also show the mutation.
So another question about VEXAS that has come in, and that is VEXAS due to loss of function of UBA1 or a new function of the mutant gene? Do either of the two of you want to speak to that question?
I can take it. So I didn't get into the exact mechanism of VEXAS. It's quite fascinating. So the UBA1 protein has two isoforms. There's a nuclear isoform and a cytoplasmic isoform. And the one intriguing clue with VEXAS is almost every one of these patients has a mutation at codon 41. So what's so special about codon 41? Well, it turns out it's the translational start site for the shorter cytoplasmic isoform. And what happens in these patients is they lose that start site and they jump to the next methionine, which is at codon 67, and synthesize an even shorter short isoform, which is catalytically impaired. So they retain nuclear ubiquitylation from the long isoform, and they lose cytoplasmic ubiquitylation from the short isoform.
Okay, excellent. Next question. Also for Dr. Grayson. Do you think that the other 92% of relapsing polychondritis patients have mutations in the ubiquitin pathway?
Yeah, a really great question. So, you know, is the ubiquitin pathway teaching us something about chondritis? Why do patients with VEXAS syndrome usually get skin disease, chondritis, and vasculitis? What is the relationship between ubiquitin and those specific tissue tropism? We don't know. I know that we have exome sequencing data on our relapsing polychondritis cohort and have looked for deleterious mutations in other parts of the ubiquitin pathway and haven't found anything similar. So whether ubiquitin plays a broader role in this disease or whether there's convergence on pathways by some other means, it remains to be seen. But certainly monogenic diseases that recapitulate more complex diseases have a lot to teach us and a lot of hypotheses to push forward.
Yes, indeed. So let's see my questions. Okay, so now we have a somewhat philosophic question, which can be addressed to any of our three presenters here. So it's a two-part question. One of the first part has to do with developing animal models for somatic mutation. And the fact, of course, is that the typical animal model system would be mice, and mice only live a couple of years. And so if somatic mutations take some time to accumulate in order to have a biologic effect, then are mice a very good model system? And then the second part of the question is thinking about Einstein's theory of relativity, that, you know, supposedly as we travel closer to the speed of light, I think that then clocks are supposed to slow down. And so would the rate of somatic mutation change if one were traveling in a spaceship close to the speed of light?
Well, I'll take a stab at least at the first, and for both, I think VEXAS and well, speaking for as a hematologist for CHIP and for PNH, animal models have not been useful. And I think that that's often a problem in immunology as a whole. I think with my immunology side, that I think we've overestimated and overinterpreted a lot of animal models, convenient as they are to publish, but they don't necessarily correlate very well globally with the human immune system, partly due to the difference in the animals, not so much the genome difference in the animals, differences in the environments that they exist in. They're really quite artificial species at the end. I think I would worry less for the second about the speed at which you're traveling, but much more about the inflammaging aspect that's distinct from classic chronologic time. And I think that the whole story with CHIP as well as with VEXAS and what's cause and what's effect, it really has highlighted, emphasized the role of just chronic inflammation, which may be very low level, but over a lifetime leads to accumulated damage, both mutagenesis as a direct effect of inflammation, but also selection of clones, as you alluded to, Peter, for example, in the inflamed colon, clones that are resistant to very specific local elements of inflammation.
And then it makes one also wonder to the next level as a rheumatologist, what is the effect of cytotoxic and immunomodulating treatments on this process? And there's additional layers to the complexity of it beyond inflammation. What does control of inflammation do?
Indeed. So here's a question that sort of comes from Dr. Grayson's presentation, but I think either Dr. Grayson or Dr. Young might want to comment on this. So the question is, Dr. Grayson pointed out that the leukocytes in patients with VEXAS look pretty crummy, look kind of sick, like they're not going to do very well. But yet, for some reason, they seem to persist. And in fact, in time, seem to be more prevalent than the normal form of those leukocytes. How can that be?
I think that's the great million-dollar question in VEXAS syndrome. How does a toxic mutation clonally expand? And I think if we understand that question, we'll understand how to better and more effectively treat the disease. But purely speculative. I mean, one could make the argument that it's a toxic mutation, but in a toxic environment, it's no longer toxic. It may have some advantage. I don't know.
So there's an interesting phenomena from modern evolutionary theory called exaptation. And the idea there is that what we see in classic evolution as something that's difficult to explain in terms of its adaptation to the environment at some point in the evolution of the animal was in fact advantageous. But we've lost our understanding of how we got from A to D because of what happened in between. And I think that that's quite applicable to somatic mutations. So UBA1 may provide a real growth advantage or survival advantage, for example, to the stem cell, but lead to a different phenotype in the mature cell that relates to inflammation and also affects the ability of the cell to survive, at least in vitro and maybe also in vivo.
Yeah, that's a very attractive hypothesis, and certainly, as Dr. Grayson said, it's something that we can be looking at, over the course of the next few months to years, to try to figure out what's going on.
Yeah, I would add to Peter, and I say, there is the $1 million question in terms of VEXAS, but there are a lot of $10,000 questions out there also. And I think that immunologists, not you guys, of course, you clinical immunologists, but I think immunologists have been very resistant to the idea that mutations, somatic mutations, actually contribute to disease. The example you gave from, the Australian group is really a terrific one, but the senior author of that paper was supposedly laughed off of a Keystone conference podium when he suggested it might be useful to look at mutations in macrophages and lymphocytes. And I think that that's where the $10,000 answers are. And the examples of Felty syndrome, large granular lymphocytosis. We've looked at data too with our NIH colleagues. So mutations that occur in a stem cell alter the behavior of a mature cell, like a macrophage or like a very specific lymphocyte that can have a mutation in STAT2, STAT3 pathway, makes it hyperactive when it meets its cognate antigen, harmless if that particular lymphocyte is never engaged. And I think if I were a lot younger and a rheumatologist, I would be out sequencing macrophages, doing single cell work in lots of diseases, looking for mutations in the known pathways. And you can look at thousands of genes, obviously, so easily with genomics, come up with those panels. And Satu, my collaborator in Finland, has been a leader in this in terms of developing these large genomic screens. And in even my disease, aplastic anemia, we reported with her as a collaborator that a substantial number of patients have mutations in lymphocyte pathways that you would predict would increase cytotoxic lymphocyte activity. And that's not something you'd think even as a hematologist that you'd want to look at, looking now at the effector cells, not just at the target cells for their somatic mutations. And lymphocytes and macrophages are supposed to be genomically unstable, right? They're supposed to be adaptable to their environment. So they're kind of prime candidates to go wrong on occasion.
Absolutely. Well, we've reached 6 o'clock, but one last question, and then I will let you guys go. So, is it necessary for cells to be dividing in order for them to develop somatic mutations? Could you have somatic mutations, for example, in the brain, where supposedly the cells have finished dividing by the time that you're into adulthood?
Yeah, let me take that. So the answer to the first is that, as I probably went too quickly, and it's a hard concept, but these cytosine demethylation occurs even in between mitoses. So that change in the cytosines to uracil doesn't require mitosis except to be expressed. And the second is that the brain grows early on, and the brain is full of mutations. And I think, a very provocative idea is that mutations that are acquired early in neurologic development, brain development, could be responsible for diseases like schizophrenia or depression, with very selective areas having functional changes. And that is known to be the case for example, some cases of refractory epilepsy, where the neuronal function has been altered by an early occurring somatic mutation.
All right. Well, thank you very much. That was really a fantastic answer. And at this point, I want to thank both of our speakers, Dr. Young and Dr. Grayson, for excellent lectures, and Mr. Furson for really putting a face on an important new disease. I thank everyone for their participation, for wonderful questions having been submitted. Well, I look forward to seeing you all again next week for the next installment of Demystifying Medicine. Have a good afternoon, evening, or morning, whatever it may be to you.
Thanks, Dan. Thanks, Wynn.
Dan, can we stick on for a few minutes?
Indeed.